and welcome to Mattermosaic. Piecing together the universe, one discovery at a time. Today, we're exploring a breakthrough that could change the course of humanity itself. Imagine being able to edit the very code of life, as easily as you might edit a sentence on your computer. What if we could erase a deadly mutation, insert a protective trait, or even rewrite evolution itself? That's not science fiction anymore, that's CRISPR. Before we dive in, don't forget to subscribe to Mattermosaic for more explorations of the discoveries that shape our past, present, and future. The blueprint of life. To understand why CRISPR is so revolutionary, we need to zoom in on the very foundation of biology: DNA. Every living thing on Earth is built from DNA, a long molecular chain carrying instructions in just 4 chemical letters: A, T, C, and G. Arranged in specific sequences, these letters form the instructions for building proteins, which in turn build cells, tissues, and ultimately entire organisms. For billions of years, DNA has been rewritten by nature itself. Mutations, tiny errors in copying DNA, created diversity. Natural selection kept what worked and discarded what didn't. That's how life evolved from single-celled microbes into trees, elephants, and us. But evolution is slow, random, blind. What if we could take control? The discovery of CRISPR. The story begins not in a medical lab, but inside bacteria. In the late 1980s, scientists studying bacterial DNA noticed strange repeating patterns, like mirrored sequences separated by unique spacers. They didn't know what they were, so they called them CRISPR, clustered, regularly interspaced, short palindromic repeats. It wasn't until the 2000s that researchers discovered the truth. These sequences are part of an ancient bacterial defense system. When viruses attack, bacteria snip out a piece of the viral DNA and store it in their genome as a mugshot. If the virus returns, the bacteria use CRISPR to recognize it and unleash a protein called Cas9, which slices the invader's DNA apart. In 2012, 2 scientists, Jennifer Doudna and Emmanuel Charpentier, realized this bacterial trick could be turned into a universal tool. By programming CRISPR with a custom guide RNA, Cas9 could be directed to cut any DNA sequence we choose. Suddenly, humanity had the power to edit life at will. For this breakthrough, Doudna and Charpentier were awarded the 2020 Nobel Prize in Chemistry. How CRISPR works. So how does this actually happen? Picture CRISPR as a pair of molecular scissors guided by a GPS system. The GPS is the guide RNA, which tells Cas9 exactly where to cut. Once the DNA is snipped, the cell's natural repair mechanisms take over. Sometimes it just seals the break, effectively disabling a gene. Other times, scientists insert a new piece of DNA into the cut, rewriting the original sequence. This means we can delete mutations that cause disease, add sequences to boost resistance, or modify organisms for entirely new purposes. It's elegant, powerful, and surprisingly simple, which is why CRISPR has spread like wildfire through biology labs worldwide. Applications of CRISPR medicine. The most immediate hope is curing genetic diseases. For example, sickle cell anemia is caused by a single mutation in the hemoglobin gene. With CRISPR, scientists can precisely target and correct that mutation. Trials are already underway, and the first patients have shown remarkable recovery. Beyond inherited diseases, CRISPR holds promise for cancer therapies, where immune cells are engineered to better recognize and destroy tumors. Some researchers even see potential for curing viral infections like HIV by cutting viral DNA out of human cells. Agriculture. The food on our plate could also be transformed. Imagine crops resistant to drought, pests, or disease without needing heavy pesticide use. Or rice fortified with nutrients to fight malnutrition. Unlike older genetic modification methods, CRISPR is far more precise, cutting down on unintended effects. Environment. Another bold idea: gene drives. By editing insects like mosquitoes to reduce their ability to carry malaria, CRISPR could save millions of lives. But these ecological interventions come with enormous caution. Once released, such changes could spread uncontrollably. Ethical questions. With such power, it's no surprise that CRISPR has sparked fierce debates. Editing genes in adult cells to treat disease is one thing. But editing germline cells, eggs, sperm, or embryos, means altering the human gene pool forever. Future generations would inherit those changes. This leads to the infamous concept of designer babies. Should parents be able to choose their child's traits, intelligence, height, eye color? That might sound tempting, but it risks creating inequality, where genetic advantages are only available to the wealthy. In 2018, a Chinese scientist shocked the world by announcing he had created the first CRISPR-edited babies, supposedly resistant to HIV. The backlash was immediate. Many argued it was reckless, unsafe, and unethical. CRISPR gives us power beyond imagination, but also responsibility. Humanity must decide not just what we can do, but what we should do. Future possibilities. Where could CRISPR take us next? Treating blindness by repairing faulty genes in retinal cells. Developing crops that thrive in a warming climate. Engineering bacteria to clean up oil spills or capture carbon dioxide. Even preparing humans for life on Mars by giving us resistance to radiation. It's a future filled with both hope and uncertainty. CRISPR is not magic. It's a tool. But like all tools, its impact depends on how wisely we use it. This is Mattermosaic, piecing together the universe, one discovery at a time. If you enjoyed exploring CRISPR and the future of gene editing, let us know in the comments. If you could rewrite life's code, what would you change first? And don't stop here. Keep the journey going by watching our next deep dive, where we uncover how quantum physics SE